Now we come to the 11th lecture in this series, which is an updated version of the stuff that we gave in our summer school course from 2003 to 2013. And this shows the results of using our fitting methods and the lessons that has taught us about the nature of partial agonists and to some extent the nature of the transitions between the various states in which both the glycine receptor and the nicotinic receptor can exist. So now we are going to discuss the mechanism which we came up with, first by the glycine receptor, but then subsequently by the nicotinic receptor also, and we applied it to partial agonists. So the first paper in which I was involved, at least, was the heteromeric glycine receptor, and this was largely the work of Valeria Bertzamato, who was then a PhD student of Lucia Civilotti's, and other people were involved too. Here's some responses to glycine. These are heteromeric glycine receptors expressed in HEK cells. With a low concentration of glycine you get something like this. That's the calibration here is 1 second, so that's quite a long record. The calibration here is 50 milliseconds, so it's expanded rather more than 20 times, and we can see short openings, fairly rare. At a higher concentration, 50 micromolar, we see openings are more frequent. The openings are upwards, of course, and the probability that the channel is open, assuming these stretches all come from the same ion channel at least, is 0.3. At 100 micromolar, the probability of being open was 85%, and at the highest concentration we tested, the probability of being open was 93%. This is fairly obviously all from one channel here. We have of course cut out these long desensitized periods on each side of the periods of activation, and that enables us to get the best of all sorts of dose response curves. The probability of being open against glycine concentration, the probability of course goes from 0 to 1, but it's not normalized to 1, and the maximum isn't in fact 1, it's about 96% here. So this looks like a good promise, a promising sort of receptor for analysis, because it's got a good strong concentration dependence. This is actually fitted with a Hill equation, which is the wrong equation, of course. No physical mechanism predicts a Hill equation, but it gives a Hill slope of 3.2, which suggests that 3 molecules of glycine may be sufficient to activate the receptor, and subsequent work confirmed that. So this is the input stage for HJCfit using this mechanism, which we'll come to in a minute. Notice that I said that it's got a good strong concentration dependence. At 0.3 micromolar one sees only very short openings. That's 100 milliseconds calibration, and this is I think a 1 second calibration. At 10 micromolar you see not only more frequent activations but longer ones. They're very bursty looking, with many short shuttings in them. And there's this strong concentration dependence, which means that we get a lot of information about mechanism from the fits. There's a concentration dependence for nicotinic receptors too. At very low acetylcholine concentrations you see more short openings, at least some of which are monoliganded openings, no, perhaps not all, but that is not a very pronounced effect. You've got to go to very low concentrations to see many, whereas the concentration dependence extends over a longer range of the glycine receptor. So we're going to use HJCfit to estimate the underlying rate constants between transitions, between the different states of the channel, the different conformations and different degrees of liganding. But before doing that, I should just go again, we've done it before, but I'll do it again, about the criteria we use for whether the mechanism we're testing fits well. It's been tested by simulating experiments. This was a nicotinic receptor model, actually, with 3 open states, a doubly liganded and 2 different singly liganded open states with different lifetimes. And we fitted the Q matrix to it and used that Q matrix to predict the distributions we would see. And we compared them with and this is one of the 1,000 simulated experiments that we did. The distribution of apparent open times is here, and the HJC distribution, which was not fitted to that histogram but was calculated from the fitted parameters, goes through it nicely. The red line here is the distribution of corrected for the missed events. So you can see there's a lot of events are being missed because the red line is the calculated ideal distribution from the fitted parameters. Likewise, we can fit shut times pretty well. Again, the solid line wasn't fitted to the histogram, but it was calculated from the distribution of apparent shut times, the HJC distribution for those. You can see we can only go up to about 3.5 milliseconds here, because any longer shut time than that would separate openings that we couldn't be fairly sure came from the same ion channel. That was the critical shut time. As well as looking at the open times and shut times, we also look at the correlations between open and shut times. We use 2 different methods for that. One is to look at conditional distributions. So this is the open time along here, is the open time for only those openings which appear adjacent to apparent shut times that are shorter than 100 microseconds. And the red dashed line is here something different from there. What it is the distribution of all open times. In other words, it's the same shape as the blue line in that part of the diagram. And you can see that compared with the distribution of all apparent open times, there's far fewer long openings next to short shut times, and there's an excess of long openings next to short shut times. In fact, we don't often do that because you can do it, you'd have to plot it separately for each open time, each shut time range here. What we usually do is just look at a synoptic plot of that, which is to take the mean open time against the adjacent shut time. We define ranges of shut times. The mean of this range is plotted along here. The mean open time next to them is plotted here. So for 100 microseconds, which is what was there, the mean of that distribution would be plotted as a blue square here, along with its observed standard deviation. In this case, the observations of the blue diamonds, they've got errors on them, but the red symbols are the values for these means calculated by the HJC method from the fitted parameters. Again, not fitted to the observations, but these are the calculated mean open times conditional on being next to shut times within the specified range. And again, they fit. Like the dashed line through them is the theoretical distribution, which is continuous because in order to get observed points you have to use shut time ranges here, but you can calculate the conditional distribution as a continuous thing. Of course it goes pretty much through the points, the red points, and that fits well too. So we say that this was a successful fit insofar as it predicted what we observe. So we start HJCfit by postulating a mechanism, and that is to some extent arbitrary. The simplest mechanism we can imagine with 3 bindings is something like this: 3 bindings to a resting conformation and correspondingly 3 open states. You might also consider unliganded bindings, but we can't see any of them, so we don't. And in this case we've postulated these binding sites on the resting conformation are all the same. So these coloured arrows just mean that we've constrained these to be the same. We only fit one, and the other ones are calculated from them. And if we do that and plot these apparent open time distributions and apparent shut time distributions at different concentrations, these are real experimental data now, but we see that the fitted distributions simply don't fit. And they don't fit the shut times and they don't fit the open times. So that enables us to rule out that mechanism. It doesn't, even when it's fitted by the method of maximum likelihood, the fit converges, it gives you values for the parameters, but when you calculate the HJC distributions, they don't fit the data. They also predict a dose response curve which is too shallow, has a slope only of 1.8 rather than 3.2 which was observed. Now, the nicotinic channel behaves, at least to a first approximation, as though the binding to one site, one of the binding sites, is unaffected by binding to the other. So we can say that the binding sites are independent. That isn't true for the glycine receptor, at least not with our initial fit mechanisms that were fit. When one site is occupied, the next binding appears to have a higher affinity. here's 3 binding sites to the resting conformation, and you can see that when these aren't constrained to be equal, they come out very unequal. That's 14,000 micromolar, 200 micromolar, and 10 micromolar. Huge increase in affinity, apparently, as the number of binding sites that are bound increases. Of course, we had openings connected to each of these, and that didn't give a great fit, so we had to add some more states, some more shut states in order to get a good fit. And we added states here which are labelled as D for desensitized, but in fact they don't account for macroscopic desensitization, because you can see that they all have pretty short lifetimes. So they're a bit mystery, these sites. They're just fudge factors, really. But that first attempt to fit it reminded us rather of what happened with hemoglobin. With hemoglobin, it also seemed that each subsequent molecule that was bound with higher affinity, as though the 4 binding sites in hemoglobin interacted with each other. Once one was bound, the next one was changed in such a way as that it had a higher affinity. This was fitted with something called the Adair equation, which consists of 4 successive bindings, each with the affinity unconstrained, and it appeared to get higher. But Jeffries Wyman in 1951, rather underestimated person in the field of receptors, I always think, had suggested that the cooperativity in oxygen binding, this increase in apparent equilibrium affinity, where there's more molecules are bound, it had been described as an interaction between the binding sites, but he postulated that it actually resulted from the fact that hemoglobin existed in 2 different conformations, one of which had a higher affinity for the ligand than the other. But for any one conformation, there was no interaction between the binding sites. And our flip model applies to this thinking to the glycine receptor too. So we have the binding of a first glycine molecule, binding of a second glycine molecule with the same equilibrium constant, a third with the same equilibrium constant. Now, if you just had them opening, of course, you wouldn't get a steep dose response curve the way we do. But if we have another shut conformation here, which we, for the sake of argument, we can call it the flipped conformation, then, if it turns out that this equilibrium constant corresponds to tighter binding than that one, then we have something very like hemoglobin. In fact, this part of it would be the mono-Wyman-Changer mechanism for hemoglobin, because mono-Wyman and Changer famously made systematic the model which Wyman had first suggested in 1951 and applied it to receptors in a qualitative sort of way. At least they couldn't actually measure it. But of course, in a simple mono-Wyman-Changer, these would have been shut states and they would have been open states, and that doesn't fit the glycine receptor. The open states come from these flipped conformations. So we're postulating that all the binding sites are independent on any one conformation of the receptor. If the receptor doesn't change conformation, then that makes a lot of sense. Why should a neighbouring binding site be affected by the occupation of one if the conformation of the receptor hasn't changed? Likewise, on the flipped conformation, the equilibrium constants for binding are assumed to be the same in the mechanism. The question is, does that mechanism fit? And you can see this mechanism fits actually very nicely. We fitted simultaneously 4 different concentrations, as we described in the last lecture. You can see that at 10 micromolar, at 30 micromolar, 100 and 1000, the apparent shut times are fitted very well by the continuous line, which is the HJCfit distribution, not fitted to that histogram, but calculated from the fitted parameters, the fitted Q matrix. You can also see that there's a huge number of missed events at this high concentration, because the ideal open time calculated from the fitted parameters is very much less than the apparent open time. That was used as an example of an extreme case where it was essential to fit to allow in the fit for missed events, because so many are missed. Likewise, the apparent shut times also fit very nicely with the HJC distributions, except perhaps this one concentration here where the fit is a bit less than perfect for 100 micromolar glycine, but even then it's not bad. These are the conditional mean open times plotted against the mean of the adjacent shut time range, and they, again, the observed curves are fitted by the theoretical prediction. Notice that the correlation actually disappears at the highest concentration, and that's what you'd expect, because at the highest concentration all you've got occupied is these 3 states with a single open state essentially, and that means if there's only one open state, then of course there are no correlations. This is the P-open curve that was observed. The points were observed. That line is not fitted to the points, but it is again calculated from the HJC type calculations, allowing for missed events. The solid line allows for missed events. The dashed line doesn't. The fraction of time for which it's open is not very sensitive to missed events, of course, but it does make a difference. They fit with when you allow for missed events, the solid line goes through the top points pretty much exactly, whereas the dashed line, which doesn't allow for missed events, misses them by a bit. So this is from the work of Valeria Bertamato. So this flip mechanism fits as well as the interaction mechanism, where the sites appeared to interact, but it gives a physical explanation of the interactions, and also incidentally it has 14 free rates rather than 18, so it's a bit more precise to fit. To put some numbers on the fits, all 3 sites on the shut conformation, the resting conformation, are quite low affinity, 520 micromolar, half a millimolar almost, and they're all the same because the conformation hasn't changed. Binding to the flipped conformation is tighter though. That's where the cooperativity comes from. It's only 8 micromolar, that's 65 times high affinity for the flipped conformation. Notice that this higher affinity doesn't come primarily from a fast dissociation rate, it comes from a fast association rate. That's only 0.6 times 10 to the 6, rather slow association rate, whereas this is 150 times 10 to the 6, 1.5 times 10 to the 8, a more typical association rate constant. Notice also that because of microscopic reversibility, the fact that the affinity for this is 65 times greater than the affinity for the resting conformation implies that these isomerization rate constants from resting to flipped will also vary by a factor of 6.5. So for the fully liganded receptor, the flipping equilibrium constant is 27. In other words, there's 27 times as much of that as there is of this at equilibrium. Whereas for the singly occupied receptor, it is very reluctant to flip. That equilibrium constant is only 0.006. So there's the amount of this compared to the amount of that is 0.6%. If you go on to the opening stages, again we find that efficacy increases as the degree of liganding increases, but we haven't joined up these not because we're asserting that they can never dissociate, but simply because they don't obey microscopic reversibility. If they did, the ratio of that to that, which is 10, would be the same as the ratio of that to that, which is less than 2. So the opening step doesn't appear to obey microscopic reversibility. This is a really interesting observation, I think, and it was predicted by Peter Leuger in the early 80s. If there is any interaction between the flow of an ion through the channel and its opening and shutting, then you wouldn't expect the microscopic reversibility to hold. Here there's no ions flowing in the resting and flipped conformations, but here there's ions flowing through, and maybe they interact with the opening and closing of the channel, and that's why it doesn't obey microscopic reversibility. That's just a speculation. This was an early speculation about what the flipped conformation might consist of, its higher affinity, and it might be thought correspond to the closure of loop C on top of the agonist binding site. But that's probably not the case, because the higher affinity for the flipped conformation results from a higher association rate constant, not a smaller dissociation rate constant. If the binding site had become capped, you'd think it was harder for the agonist to get in and harder for it to get out. So the association rate constant should be decreased and the dissociation rate constant should be lower. There was a sort of dictum in enzymology, at least, that the affinity for binding sites was controlled largely by the dissociation rate constant, not the association rate constant. So if something was tightly bound, it was because it was bound for a long time rather than because it associated easily. But in these receptors, the opposite seems to be the case, and that's the case for nicotinics as well as for glycine, as we'll see. Quite how it is that the higher affinity is caused by a higher association rate constant isn't really understood in structural terms, but then actually remarkably little is stood in structural terms despite all the hype about it. We pointed out last time that once you've got the Q matrix you can calculate anything, and so we ought to be able to calculate the time course of the response to a brief pulse of agonist. And insofar as the synapse releases glycine for only a short time, then that we can, maybe a miniature synaptic current can be, the time course of that can be calculated from the mechanism. So here's some glycinergic inhibitory postsynaptic currents recorded from spinal motor neurons by Marco Beato. There's a 5-second sweep showing a lot of miniature synaptic currents. Here's a much enlarged one, 500 millisecond calibration. And if you average a lot of these, or superimpose a lot of them, then you get something like that. And when you average them, you can get quite a smooth synaptic current time course. And this is the time course fitted by our first model we tried that didn't work very well. 3 non-interacting binding sites doesn't fit at all the synaptic current. In fact, none of those do. But if we calculate the time course of the current that, or P open, in response to a brief pulse of agonist such as might be released at the synapse, we get a line which goes pretty much through the observed average synaptic current. The continuous line there is the predictions of the flip model. The slightly noisy one is the average synaptic current. And that's not, the fit isn't perfect, but it's pretty good considering the very different conditions under which these measurements were made. The predictions were calculated from steady-state recordings of heteromeric glycine receptors in spinal neurons, in HEK cells, recombinant ones, whereas the miniature synaptic currents were native glycine receptors in the spinal neurons. So that's pretty good in the circumstances. So, having satisfied ourselves that the flip model was a reasonable description of the synaptic currents and of the observed single channel currents in the glycine receptor, we thought, what is different in a partial agonist? Because the maximum response is controlled by 2 things now, not just an opening and shutting reaction equilibrium constant, as in the case of the Castillo-Katz mechanism, but is also dependent on the equilibrium constant for flipping. So which of these is different, we thought, in a partial agonist? And this led to our 2008 paper, the work of Remigius Lappe, who was working by then and in the group of Lucia Civilotti. I'd already retired by then, nominally. and this worked quite well. The first partial agonist we looked at was taurine. Here the openings are upwards again, and you see that with a millimolar glycine the channel is open for almost all the time, but with a saturating concentration of taurine it's only open for about half the time. And if you plot the P-open curve, these marvellous dose response curves on an absolute scale, not relative, it goes up to almost 1 with glycine, but not quite to 1. That's about 96% in fact, whereas with taurine it's about 50%. These are a couple of the brief shuttings on which so much depends. That's a 115 microsecond one that reaches almost to the baseline. This is a 30 microsecond one, which was the resolution we quite often use, so we deem that to be real. At least if it was a quiet recording, you can pretty reliably tell that that's a real shutting, not just noise. So we've already fitted the glycine receptor with the flip model. Does it fit? Does that same mechanism also fit taurine? So this is the flip model we used for glycine. In this case we couldn't detect singly liganded flippings or openings, so that bit of it's missed out, not because it's not there, but simply because we can't detect it. And the result is, these are for apparent open times. Again, the solid lines are the HJC distributions calculated from the fitted parameters, the fitted rate constants, all these numbers on the arrows. They aren't fitted to the histogram. But at all three taurine concentrations that were fitted simultaneously, 3 millimolar to 100 millimolar, you see that they fit well. And the same was true of the apparent shut time distributions. They all fit well over the range where they were determined. And the P-open curve is also fitted reasonably well. Again, this line was not fitted to those points, but it was calculated from the fitted parameters, so the fit is a good test of whether the mechanism is a reasonable one or not. And the outcome of this was that taurine is a partial agonist because it can't flip, not because it can't open. So if you look at the equilibrium constants for glycine in red, taurine in blue, we see for the fully saturated receptor, we're not dealing with all of the results here, but you can find all of the results in the 2008 paper. We find those two equilibrium constants for glycine and for taurine on the resting conformation. They don't differ that much, in fact. Whereas for the flipped conformation, the affinity is 65-fold higher for glycine, but only about less than twice as high for taurine. If we look at the equilibrium constant for the flipping reaction, it's 27 for acetylcholine, so this concentration, that is 27 times the concentration of that at equilibrium, but it's only 0.15 for taurine. It has a much lower tendency to flip, 180-fold lower in fact. If you look at the efficacy step, the opening reaction, they don't differ by 180-fold, but only by about 2-fold, so the gating is similar. And it's interesting that the reason for the higher affinity of glycine for the flip conformation, it depends on the association rate constant, not on the dissociation rate constant. The association rate constant for glycine here is 63 times faster than that for taurine, and that accounts almost entirely for the greater affinity for the flip state. If we again look only at the saturated receptor, then we get we can only have 3 things to consider: the resting conformation, the flip conformation, also shut, and the open conformation. For glycine, it only spends 48 microseconds in the resting conformation on average. For the flip conformation, it's even briefer, 7.7 microseconds, and it spends about 5% of its time in the flip conformation. We also see that there's a very fast channel opening rate, about 130,000 per second. That dominates the lifetime of the flipped state, so 1 over 130,000 is about 7 microseconds. And at equilibrium, 95% of the time the channel will be open according to these numbers. Whereas for taurine, the flipping reaction is much slower. The unflipping reaction is somewhat faster. So because of this slow flipping, the lifetime of that state is much longer. It's 1.3 milliseconds. The flipped state is again about 7 microseconds average duration. The open state at equilibrium, it's in for 50% of the time. I mean, that's because it's a partial agonist. One way of looking at the equilibrium constants for that is to take their logs, which can convert to something proportional to the free energy changes. And that shows again that the partial agonism rests in the flipping stage, not in the open and shut stage. This is the resting conformation here. This is the flipped conformation there, and that's the open conformation there. There's energy barriers between these 3 conformations. And for glycine, it's a downhill transition to the flipped state. In other words, it would rather be in the flipped state than in the resting conformation. This is for the fully liganded receptor, remember. Whereas for taurine, it's an uphill change to the flipped conformation, but once it's in the flipped conformation, the opening and shutting reaction is much the same for both of them. The overall change in energy from the resting conformation to the open one is very downhill for glycine, whereas for taurine it's almost on a level. The energy of the fully liganded open channel is almost the same as for the resting conformation, which is why half of it's open and half of it's shut. Okay, that's it for glycine. Now let's go on to the nicotinic receptor. It was, after all, my first love. Acetylcholine is a pretty good agonist. It opens the receptor for 97-98% of the time even when it's saturated. Tetramethyl ammonium, on the other hand, is not such a good agonist. This is the p-open curve for tetramethyl ammonium. It reaches about 70-odd%, even at the highest concentrations. So is that because of a deficiency in the opening reaction, or is it because of a deficiency in the flip reaction? So we fitted this flip mechanism for TMA and for acetylcholine. Of course, there's only 2 binding sites in this case, not 3 as for glycine. And we got good fits for the apparent open times at both concentrations and for the apparent shut times at both concentrations. At this lower concentration, we could only go up to 0.1 milliseconds because otherwise you couldn't be sure that the subsequent openings all came from the same channel. But it fits over the range that was used. So that looks good. And what was the outcome? And the result was that tetramethyl ammonium is partial because it can't flip, not because it can't open, just as in the case of glycine and taurine. Again, we consider only the saturated part of the curve, so high concentrations. The efficacy for the gating reaction was very similar for the 2 agonists, acetylcholine and TMA, 34 and 28. Whereas for the flipping reaction, there was a 27-fold less tendency to flip compared with for TMA compared with acetylcholine. The equilibrium constants, for example, for acetylcholine in red, TMA in blue. We only consider the saturated part of the curve, what you get at the saturated part of the mechanism, what you get at high concentration. The resting affinity for acetylcholine was 40 micromolar. It was much lower for TMA, 3000. The actual opening reaction has an equilibrium constant which is very similar for both of them. Whereas the flipping reaction has a much lower tendency to flip for TMA than it has for glycine, 27-fold lower, and that's the ratio of those 2 equilibrium constants. And why is this difference between the binding rates, the binding affinities for the 2 agonists so strong? Well, the association rate constant for acetylcholine was 271 times 10 to the 6, 2.7 times 10 to the 8, in other words, a typical fast association rate constant. That's molar to the -1, second to the -1. Whereas the association rate constant for tetramethyl ammonium was much slower, only 5.6 times 10 to the 6. Whereas the dissociation rate constants for both of those agonists were much the same. Again, we see the strong effects tend to be on association rate constants rather than dissociation rate constants. So, why does flipping matter? No pun intended. Let's revert to the very simplest reaction we had for any receptor of the Del Castillo-Katz mechanism, a binding step followed by an opening reaction. The maximum opening reaction was depended on the equilibrium constant for the opening reaction, so it was E over 1 plus E. That's just a conformation change there, of course. So what difference does it make if we flip? Well, again, if we're considering only the saturated receptor, we can make some sort of approximation just by putting an extra flipping step in the Del Castillo-Katz mechanism. So it now has 4 states, not 3. Of course, only one agonist is bound rather than 3 for glycine. So this is an oversimplification, but it's very instructive. In fact, at equilibrium, this mechanism is indistinguishable from the simple Del Castillo-Katz mechanism. In this case, the maximum P open was E over 1 plus E, and in this case, you can also express it as E over 1 plus E if you define the effective E as that, because the actual expression for P open maximum in this case is that, and that can be written as E effective over 1 plus E effective if you define E effective as that. It's the actual equilibrium constant for the opening reaction divided by the fraction of shut channel which is in the flip state, which is F over 1 plus F. In fact, not only is it identical, indistinguishable macroscopically from the Del Castillo-Katz at equilibrium, it can even be kinetically the same, because if the flipping reaction is fast, if that were the case, then this would behave kinetically like one state, the shut but occupied state. But the effective opening reaction rate constant would be the fraction in this state, the fraction of things, of receptors in this compound state that were in the flipped conformation, because it can only open when it's flipped. So if this equilibrium constant is very much over in that direction, then the estimate of the opening rate constant won't be much affected, because most of this will be in the AF state. Whereas if it's mostly in the AR state, a poor flipper, then not much of it will be here and therefore available to open. Just consider a numerical example with a K value of 100 micromolar and an F of 10 and an E of 10. Well, 10 is quite large compared with 1, so the effective K value will be reduced by a factor of 11, so it'll be 9.09 micromolar rather than 100 micromolar. Whereas the effective E value, it's multiplied by 10 over 10 plus 1, so it'll be 9.09 when the true value is 10. So if F is large, the main effect is to slow the apparent dissociation rate by a factor of 1 plus F, whereas the error in the opening rate is quite small, and conversely if F is small. So let's have a synopsis now for the nicotinic receptor. Again, we're considering only the fully liganded case, resting, flipped and open. The flipped state again has a low lifetime, about 10 microseconds in every case for all three agonists. So there's only 3% of it present at equilibrium at a saturated concentration, 2% for TMA, 1% for choline. Of course, this means it's very difficult to detect structurally, because only if you use a large agonist concentration, not much of it's in the flipped state, and that's because it very rapidly leaves the flipped state for the open state. If you consider the flipping reaction alone, then we see that the forward flipping reaction is what changes most between these different agonists. The forward flipping reaction is 22,000 reciprocal seconds for acetylcholine, but it's only 220 for choline, intermediate for this. The unflipping reaction also gets a bit faster for the partial agonist, but not by as much. Whereas the forward flipping rate, the forward opening rate, these are the open state here with the asterisks, is always very fast, 87,000, 71,000, 52,000. And that's not quite as fast as for glycine, where it was 130,000, but it's a similar ballpark. The shutting reaction changes very much less. It's 2,000-ish for all of those. In other words, about the individual openings last half a millisecond, but it'll oscillate a lot of times between these two states to give a long activation, a long burst of openings. So the effective opening rate is very much lower for choline than it is for acetylcholine, but that difference lies largely in the flipping reaction. In fact, if we draw a diagram like the one before, resting, flipped and open, we can see that for acetylcholine it's downhill to the flipped state, it's downhill again to the open state, so the overall reaction is very much downhill, that red arrow there. For TMA, tetramethyl ammonium, it's uphill to the flipped state, that's that blue arrow there, but then on it's downhill to the open state. For choline, it's very strongly uphill to the flipped state, that's that green arrow, but again downhill to the open state. And the point is that this, for the actual opening and shutting reaction, these curves virtually superimpose. All the difference essentially lies in the tendency to flip, none of it in the tendency to open. Now you may say, how can we be sure that these fits work? We're dealing with some very fast rate constants, and that's something we looked at in 2003 when the HJCfit program was first fully operational, and we did it by simulating experiments. If you've got a mechanism and you've got rate constants, it's easy to generate exponentially distributed random variables and to simulate an experiment that would be expected to be generated by that mechanism. You get variable observations, you fit them, and you get estimates of the equilibrium constants. But since they're simulated experiments, not real ones, it's easy to do that 1,000 times. We did that systematically in this 2003 paper, We also did it in the 2004 glycine paper, the Bertsamato one, because we said estimates for the opening rate constant beta 3 for the flip mechanism is very fast, 130,000 reciprocal seconds for the heteromeric receptor. At first we were deeply skeptical about our ability to estimate such a fast value from data with a resolution of 30 microseconds, because if you remember, this implies that the mean lifetime of the flip state is, well, less than 1 over 130,000, which is around 7 microseconds. So we also did simulations of the glycine mechanism, this using the rate constant which it estimated in the Bertsamato paper. And what these show, all the simulation papers show, is the distributions of the values we get by fitting the rate constants. And what we're fitting, remember, is the underlying rate constants, the shutting rate constant alpha 3 and the opening rate constant beta 3 for the fully liganded receptor, for example. Or this one, which is the dissociation rate constant from the flipped channel. And what we get at first was results that weren't so encouraging. Of course, the advantage of simulating them is that you know the answer in advance. You put the rate constant into it. So we knew that beta 3 was 130,000, but we purposely gave it a terrible initial guess, 15,000. That's even slower than the guess we'd have made for a nicotinic receptor in the early days. And we found, when we simulated a lot of experiments, the distribution of the estimates of beta 3 we got looked like this. The red arrow marks the true value. So the majority of values were the true value, but some of the repeated simulated experiments gave values which were too big. And likewise, for the shutting rate constants, we found some fits gave numbers that were looked about close to the true value, which was about 7,000 in this case, but other ones gave you values of the shutting rate constant which were too big. Likewise, in this dissociation rate constant, we found again 2 peaks. However, if you look at the estimates for beta 3, when you exclude those experiments in which alpha 3 was in the high peak, you see that they cluster very closely round the correct value, 130,000. And if you use a similar exclusion to get the distribution of this equilibrium constant, you again find a reasonable dissociation rate constant estimate. It's a bit biased to low values. The red arrow shows the true value, but it's not too bad. You might say we can distinguish the fits in that peak from that one because there's one of them's going to fit better, but in fact if you plot the maximum likelihood against the value of alpha 3, we find most of them here, but some of the fits were in the higher peak, which gave a value too high. This is meant to be about 7,000, remember. So some experiments were giving values over 12,000, but the likelihoods were absolutely indistinguishable. So the fits are the same. This is, in fact, is very reminiscent of what we discussed in the lecture on correcting for missed events, where we found that there were two exact solutions to the missed event problem in the very simple case where there was only one open state and one shut state. These look very much like two fits that are equally good in this much more complicated case. However, if we give guesses that are much more reasonable, in this case, for example, we gave an initial guess for beta 3 of 160,000 when the maximum likelihood value was the actual true value was 130,000, then we just get single peaks, and the red arrow again shows the true value. For these dissociation rate constants, we get pretty good estimates too. They're more scattered and they're somewhat biased, but they're not too bad at all. In fact, despite the strong positive correlation that we talked about between the estimates of alpha 3 and beta 3, they're among the best determined parameters, and we were worried about whether we could estimate 130,000 for beta 3. Given that the flip state was only lasting 7 microseconds and our resolution was 30 microseconds, we wondered whether we could estimate beta 3, and we see not only can we estimate it, but it's the most precisely estimated of any of the parameters. So that was gratifying. One more thing, this is about my last contribution to this area, 2012 in the Journal of General Physiology. We used it to point out some of the things you could do with the fitted parameters when you got them. One thing that can give you an insight into what's going on is to calculate the time course of the concentration of the various species. The occupancies of the various species, P1, P2, P3, P4, up to the pK. In the case of the flip model for glycine, we've got 10 states, to be state 10. We can calculate that by this very simple matrix equation, and that enables us, once we've got the estimate of the rate constants in the Q matrix, to calculate things like the response to a pulse of acetylcholine of 1 millimolar, in this case for 2 milliseconds, and then the concentration steps back to 0. And we can calculate the time course of all 10 different states in that case. The P open is actually this dashed line there. That's what you actually see, the fraction of time for which it's open. You can see that it's the solid line here is the triply liganded open state, and the P open is bigger than you'd expect from that, and not quite the same shape at the end of the pulse. And it's bigger, we can see, because doubly liganded open channels, this line here, make a substantial contribution to the final outcome, and you can see the time constants for all the others. This is the bottom 10% of that diagram blown up, so you can see some of the minor species. Another thing you can do is get a handle for which route the receptor will take through these various states if you give it a sudden pulse of agonist. And you can do it with these probabilities that are labelled on the arrows here. If we consider this triply liganded resting conformation, for example, there's a probability of 0.96 that the next transition will be this way rather than that way, and 1 minus that, 0.038. that the transition will go this way next. In fact, it's these probabilities that you use for generating the simulated experiments. We know the lifetime of each individual state. We can generate exponentially distributed values with that mean lifetime, and then we choose the next state that it goes to on the basis of these probabilities. This is simply the rate constant here divided by the sum of the two rate constants for leaving that state. So we can calculate the probability of this route through the channels by, well, we can start from here because everything starts from here if you begin from the resting state. So we go 9, state 8, state 6, 9, pi 8, 6, 8, 5, sorry, pi 5, 4, pi 4, 3 to get to the open channel. Likewise, you can calculate the probability of going that way, and you can calculate the probability of going that way. So that's a smaller probability that way, but the probability that it flips when it's diliganded, or that it flips when it's triliganded, was similar. This is for a very low agonist concentration. Of course, it'll rarely go straight through these, but it'll oscillate as it goes. So this is a simple mind-day calculation, but it can be quite informative. At a high glycine concentration, you've got that for when it flips, when it's triliganded, that when it flips when it's diliganded, monoliganded, which is about the same, and this for flipping when it's diliganded. So this is much more probable than the others at the high agonist concentration. So what can we conclude about the flipping mechanism? The maximum response depends on both flipping and gating reactions, not just on one efficacy equilibrium constant. And the open and shut reaction is very similar for partial and full agonists. The gating efficacy, you might call that, the flipping efficacy is lower for partial agonists. Ever since Del Castillo and Katz in 1957, it's been supposed that partial agonists were deficient in eliciting the open and shut transition. But our interpretation places the root of partial agonism earlier in the chain of events that follow binding than has been supposed up to now. We put it there rather than in the open-shut reaction. This makes a lot of sense when you come to think of it. We'd already found out 30 years ago now that the conductance of an open channel is the same regardless of which agonist opens it. And it seems now that the open state is constant in more ways than just its conductance. The mean lifetime of the open state is much the same whether it's opened by a full agonist or a weak agonist, and the opening rate of the channel is much the same for all agonists. So the open-shut reaction appears to be a fixed, constant process for all agonists. What differs from one agonist to another is its ability to enter the intermediate flip state from which opening can occur. And this makes structural sense, because what is it that's going to vary with the nature of the agonist is going to be something at the binding site. The opening reaction is remote from the binding site, so we wouldn't expect that it would be very sensitive to the structure of the agonist. In fact, if we do an Auerbach type phi analysis, we find that it's close to 1, especially for the nicotinic receptor, which means that the flipping reaction takes place near the binding site, which is exactly, of course, what you'd expect. Finally, I'll leave you with the most ambitious maximum likelihood fit we did, and this was for the NMDA receptor. This was done by Stephanie Schorger in 2005. There we have two different agonist binding, glutamate and glycine binding. We assume that these binding sites are independent and constrain the rate constants in that way. Here we have full occupancy by both glutamate and glycine. An isomerization reaction. This is a flipped state. We didn't call it that at the time, but we should have done perhaps. And it can open from there. It can also open from here. So we got good fits to the open and shut time distributions of that, but what does it predict for the time course of macroscopic events? Well, this is the calculated time course for the response to a 2 millisecond pulse to 2 millimolar glutamate. Again, it's meant to mimic a synaptic current. If you show the first 40 milliseconds of the calculated response, you see that it reaches a maximum after about 25 milliseconds. That's in response to this very short pulse. If we show the calculation up to 400 milliseconds, the pulse is very short indeed, and the response is about maximum around 25 milliseconds and then decays quite slowly. And we haven't superimposed this on the famous Wiley 1996 patch, but it is qualitatively very similar to what was proposed there. So what was measured, I should say, in the Wiley 1996 wonder patch, that was dealt with in the previous talk about single channels after a jump in concentration. Here I should acknowledge, I should have done after every talk, other people that have been involved in the experimental work in particular. Mainly, the main person I should acknowledge is Lucia Civilotti. She's succeeded in my job, the A.J. Clark Chair of Pharmacology, and kept the single channel business at UCL alive after I'd retired in 2004. What's really amazing is that she'll be retiring soon. I can't believe that. We should also acknowledge the enormous role played by Remigius Lape, our wonderful Lithuanian physicist postdoc who did a great deal of the work on the glycine and the nicotinic receptor. Valeria Bertsamato, who was Lucia's PhD student and who did much of the work on the 2004 paper, and Marco Beato, who also contributed to that work. Okay, folks, that is it. 11 lectures, a lot of hours of work. I do hope that I've managed to persuade at least a few of you that matrices are fun and not as difficult as you thought. Go home and have fun.